Okay. So photosynthesis in higher plant. Photosynthesis have two important steps. Photosynthesis means food making process. This photosynthesis have two parts. One, for
So we
food.
will write it, not everything.
Yeah, better you note down everything because I will explain in the shortest format. So in photosynthesis, we have to make food. Food means glucose C6H12O6.
सर
Correct?
सॉरी टू इंटरप्ट यू मैंने सुना नहीं आपने बोला लिखना है नहीं लिखना है
नहीं लिखना है
लिखना
क्योंकि
है
ये शॉर्टेस्ट फॉर्मेट में मैं एक्सप्लेन करूंगा.
ठीक
तो फोटोसिंथेसिस में वी नो द बेसिक प्रोसेस कि we will consume CO2. CO2 react with the water in presence of sunlight and chlorophyll. Sunlight and chlorophyll, and ultimately food will be produced plus H2O plus oxygen. Correct? This is the ultimate process. Now we want to understand this in the more detail, and to understand more detail how this oxygen is produced and how food is produced. For oxygen production where light is involved, we study about the light reaction, involvement of light. Involvement of light. So what happen in the light reaction? First, light absorbed. After light absorption, it will interact with the chlorophyll. Water will split. If
गुड
water
मॉर्निंग
split, good morning, Pratigya. If water split, oxygen is produced. It's okay, Pratigya. Oxygen produced and ATP and NADPH are produced. Why we want to produce this ATP and NADPH? You know, formation process require energy, correct? And breakdown process release energy. Example, respiration process is a breakdown process, catabolism. It release energy. But food synthesis is an anabolic process. You require energy. How you will get energy? Correct. Example, when we breathe, we take in oxygen, we produce energy, respiration process, right? In respiration process, we produce energy. In photosynthesis process, we need energy. How will get this energy? Light ray. What are the form of energy? ATP and NADH. These ATP and NADPH will be utilized in the dark ray. That's why because it is not directly dependent upon the light, so dark reaction or light independent reaction. Correct for biosynthetic phase. Biosynthetic phase. Right. Why? Because actually there is a synthesis of important part, biosynthetic phase. In this light reaction, there are multiple things happen. Multiple thing like first, there will be, I would say, ki electron transportation. How electron are transported in case of plant. And second, due to the electron transportation, how ATP and NADPH is produced. So we read about two, three topics. One, cyclic photophosphorylation, non-cyclic photophosphorylation, chemosynthetic uh, chemosmotic hypothesis. Right. Chemosmotic hypothesis. How electron transportation is responsible for this. So when we trap the light, we need some system to process the light. And for this we use word photosystem. Photosystem to process the light. Where is Jordan, Anvita?
I'll call him.
Jordan and Sarah. Yeah, call Jordan and Sarah both. So, photosystem are two types, photosystem 1 and 2. This is based on their discovery. Key photosystem 1, elder sibling, and photosystem 2 is older sibling. Correct? Older, sorry, elder and younger sibling. Correct? So, elder one will have more potential. Photosystem 1, you will see कि it can absorb maximum 700 nanometer light, but young one can process minimum energy, that is 680 P680. What does it mean? If you have light up to 680 nanometer, then photosystem 2 can process it. Photosystem 1 can also process it. But if you have light, okay, by the way, this is interesting. If you have light like 640 nanometer, both photosystem 1 and 2 will act and they will be responsible for the energy. If you have 680 nanometer, both 1 and 2 will work. If you have 690 nanometer, only photosystem 1 will work. And if you have 720 nanometer light, no one will work. Correct? Understood this part? No one will work at 720. Why? Because wherever we write this P700, this is the maximum value. This is the maximum value.
Good morning, sir.
Okay, so
Joining in.
good morning, Jordan. So this was really, really important. Photosystem is responsible for the trapping the light, and for trapping this light, there are elder one can trap 700 light, and younger one can trap 680 nanometer light. Next, in this photosystem, there are two parts. One is reaction center, and second is the light harvesting complex. This light harvesting complex is also known as antenna. You know how we used to get antenna in our houses. It will trap all the light. Light harvesting complex will trap all the light, and this will be sent to the reaction center. Reaction center is the head where everything is working. In this reaction center, you have one molecule of
Can
chlorophyll
you
A.
repeat what you said?
Yes, Rayan.
So can you again repeat what did you say just now? Didn't get it.
Yes. So we said photosystem is the main thing present in the leaf, chlorophyll, correct? Where we will process the light. So this photosystem made up of two components. Majority of the component, most of the chlorophyll A, B, carotenoid, xanthophyll, everything will form light harvesting complex. As the name suggests, it will harvest the light. It will absorb maximum light. And second, reaction center, where you have only one chlorophyll A, which is actually responsible for all the work. Which is main center part, head part. Any doubt? Anybody about this? So
So what is this exactly?
we have leaf. On this leaf, light will fall. When light fall on this, then the individual cell that contain chlorophyll, so this will absorb the light. Once it absorb the light, what will happen? There will be some electron transport, and this electron transport responsible for the energy formation. This energy later on will produce ultimately what? Wood. Makes sense? That is a summary of this topic. So in this light reaction, we want to understand how we trap this. For trapping, we have photosystem, and there is a head part, chlorophyll A molecule, head part known as reaction center, which will decide how light will be processed. Any doubt? So, anybody have doubt
One
in
second,
this?
can you scroll up?
Sure, Tanvi.
So what is this antennal?
Antenna. Antenna is the another name for light harvesting complex. The role of antenna is to absorb maximum light, because once maximum light available, only then we can have efficient photosynthesis.
Done, sir.
Yeah. This part is extremely important. And if examiner make difficult question, tricky question, they can make from this. When I said 700 is the value for the elder one, because elder one can do more work. That's why maximum potential 700. Imagine it can also work below that. Younger one have maximum potential 680 because less experience. The maximum potential is 680, but below 680 both can work. Below 680 both can work. Between 680 to 700 only elder one will work, and above 700 no one will work. Very, very, very important. Understood? And here you might get a difficult question from this part. Come to the next part. Now we know electron will be transported, and due to the transportation of electron, there will be ATP and NADPH formation. How? That is explained by non-cyclic photophosphorylation. Non-cyclic photophosphorylation. What is the meaning of phosphorylation, Aditya? ATP formation, and photophosphorylation means ATP formation in presence of light. In presence of light, if ATP is produced, the photophosphorylation. Non-cyclic, as the name suggests, that ki, in case of non-cyclic, there will be not a cyclic process. So, guys, in this case, what will happen? This is also known as Z scheme. Though it look like N, but if you see based on the oxidation and reduction, you you will observe like what should I say? Based on the oxidation reduction, it will look like this. That's why this non-cyclic photophosphorylation is also known as Z scheme. And why we call it non-cyclic? Because cycle is not formed in this case. How Z scheme is formed? Based on the oxidation and reduction. So, Anvita, what is oxidation and reduction based on the electron loss or gain?
So gain of electron is oxidation, right? I think. I keep getting.
Loss of electron is oxidation, gain of electron is reduction. Correct?
Okay, so
Just imagine you have any atom and understand photoelectric effect. This is the outermost electron. When light fall on this, what will happen? This electron will come out. Photoelectric effect, correct? From dual nature of matter. So if electron come out, this atom lost electron, so it will undergo oxidation, correct? It will undergo oxidation due to the photoelectric effect. Example, you have sodium. So if light fall on the sodium, what will happen? It will lose electron and it convert into the sodium positive.
yes,
Any
sir.
random example. So what is happening? Oxidation. This is oxidation, correct? If you have electron acceptor, example NADP, this will gain electron ultimately in the whole process. So it will convert into NADPH and S positive. Also, there is the addition of hydrogen, gain of electron and addition of hydrogen. So it is reduction. Okay, so you should know where oxidation happen, where reduction happen. Electron transportation occur because when light fall on individual atom, electron get excited, this electron will move, and this electron movement we are talking about. Understood? Anybody have doubt in this?
No, sir.
No, sir.
So let me ask, how is it going, guys, as of now? All of you understood everything? Raise your hand.
Yes, sir. Yes, sir.
1, 2, 3, 4, 5, 6, 7.
Sir, I don't know where the option is for me.
Okay. So Zoom user already said Rishikesh Manan. Okay. So come to the process. What will happen? You have light harvesting complex. Light harvesting complex, as name suggests, it will consume all the light. When light fall on this light harvesting complex, then electron will get excited. Electron get excited. Electron will have high energy. Electron move from low energy to high energy, low to high energy. Correct. So oxidation will happen, and you have photosystem 1 or photosystem 2 in this case. Answer will be photosystem 2. I told you story of key. You are two siblings. If you are the elder one, you will send your younger sibling to get the pizza if you order pizza. Correct? So younger one will pick up the pizza, will eat some slice. Example, there are total 6 slices. So he or she will eat at least one or two, and then remaining will be passed to you. So when your sibling is eating one or two piece, they will get some energy, but you will get more energy. That's why, like PS2 is your younger sibling. Get the pizza order, eat some slice. When they eat some slice through the electron transport system, they will get some energy in the form of ATP. Okay, out of six, imagine two is consumed by him or her. Then again, light will also fall on another light harvesting complex simultaneously. So this electron will also get excited to PS1, like pizza reach to you, and your hunger NADP convert into the more satisfaction level, more energy level, NADP. Right? As you can say, it look like N or Z. That's why jet ski. Okay. If question asks what happened in the non-cyclic photophosphorylation, ATP is produced, NADPH is produced. How many system work? Two system work, PS2 first, PS1 second. But Jordan, what is the source of this electron? The electron are moving. What is the source of this electron?
So it's from the water splitting up, sir.
Very good. So water splitting is really, really important. Water splitting is like your mother. So who is ultimately giving money? Your parents, correct? So when H2O is present in the leaf and light fall on this H2O will break down into two H positive electron, correct? And O2 will be produced. So in this process, you get your first product, that is oxygen. पहला product मिल जाएगा हमें, that is oxygen. Oxygen will come out. So if you want to understand what is the benefit of light reaction, three benefit. One, oxygen directly. Second, ATP and NADPH for the glucose formation in the next step. Then, okay. So water splitting is associated with PS2. Okay, water splitting is associated with PS2. Water splitting is associated with the PS2. Very, very, very popular for me. And both these things are present in grana. Both are present in grana, or you can say thylakoid. This complete non-cyclic photophosphorylation. Non-cyclic photophosphorylation present in grana or thylakoid, where you are producing oxygen, NADPH, and ATP. But sometimes just imagine that there is only single child. Correct? There is only single child in the house. If single child in the house, they don't have anybody to pick up, and maybe their sibling went out to the relative house. So though you fight with your sibling, but you always have fun with them. Correct? Now you don't want to eat pizza with so much eager, because वो है ना, किसी को चिढ़ा के खाने का अलग ही मजा है. Once you tease your sibling and then you eat, that's the another satisfaction. How many of you can correlate with this? Tanvi.
Sir, I didn't understand what you said. What do you say?
Like when you tease your sibling and then you eat, that is another level of satisfaction.
Eat or hit, sir?
No, no, eat.
Sir, she doesn't let me eat if I hit her.
No. Eat, E-A-T, eat. Like when you tease your sibling and then you eat, it is another level of satisfaction.
Eat what's of food? No.
Yeah, of course.
So that's what she doesn't let me eat. She'll just take my plate and
So
go.
she's the younger sibling, that's why she's never experienced it.
Yeah.
Yeah. So I should ask the elder one, Jordan.
It feels good, sir.
Like when you tease them and then you eat everything.
Yeah,
But if you are alone, you don't feel like you want to eat more because you don't enjoy the process
yeah,
that much.
you don't want to share also. Either
Ye-
ways.
Yeah. So same way, when light fall on the light harvesting complex and there is only one child, so one child will be obviously photosystem one. Then electron will get excited. Electron will be picked up by photosystem one. Photosystem one will get some energy because electron transport system. You produce ATP in this process, and ultimately again electron will come back to the light harvesting complex. This is known as cyclic photophosphorylation. As you can see, continuous cycle is formed. What
So
is
could you
happening in
repeat
cyclic?
this?
Yes, in cyclic, what happened, Rayan? Only ATP produced, only photosystem one involved, correct? No water splitting, and this whole process occur in the stroma lamella, not in not in thylakoid or brain. Stroma lamella, correct? No NADPH in this case found, no water splitting happen, and this comparison is really, really important. What are the difference between cyclic photophosphorylation and non-cyclic photophosphorylation? You want to ask, sir, what is the purpose? Why we have two different cycles, non-cyclic and cyclic? Why we have two? Why not one only? Answer is you have to because you have to balance in the next step. Example, in for one glucose formation, total 18 ATP is consumed and total 12 NADPH is consumed. Correct? In non-cyclic photophosphorylation, equal amount of ATP and NADPH is produced. So 12 ATP and 12 NADPH will be produced. Make sense? From where we will get the remaining 6 ATP? Cyclic. This one cyclic. Correct? If you see in one C3 cycle, there is 3 ATP consumed and 2 NADPH consumed. So in non-cyclic photophosphorylation, 2 ATP and 2 NADPH will be produced. What about the third ATP? From where we get third ATP? This one only, cyclic only. Make sense? Everybody understood why we need? Because you need lot of ATP for actual glucose formation, and non-cyclic is not sufficient. That's why elder one need to do extra job to get the requirement done. Understood, guys?
So how many cycles are required then?
How many cycle of what? Cyclic or non-cyclic?
So both.
Example, for one C3 cycle, you need one non-cyclic, one cyclic. For one C3 cycle, one Kelvin cycle, you need one, what should I say? One non-cyclic, one cyclic. Any doubt?
No, sir.
Yes. So tell me, anybody have question from this or you understood really well? Raise your hand.
So that C6H12O6, the whole which I've written, it is the full like 18 ATP and 12 NADPH are required.
For full glucose, na?
Ha.
For full glucose, 18 ATP and 12 NADPH. So equal amount you get from the non-cyclic. What about the cyclic part? What about the remaining people? Ryan, you understood everything?
So just try and wait a minute, sir.
Aditya, Manan,
Yes, sir.
हैंड रेज करो मुझे कैसे पता चलेगा जल्दी जल्दी निर्जला ओके कौन
Then
बचा
so it
मनन
turns out to be like a cycle wise, right? In one cycle, that 3 ATP and 2 NADP is required.
exactly for one C3 cycle one Kelvin cycle we need 3 ATP and 2 NADP so 2 ATP we get from the non-cyclic correct 1 ATP we need from the cyclic that's why we need cyclic.
Okay, sir, got it.
Okay. So as of now what we understood when light fall on the leaf oxygen
Can you
produced.
Oh, one
scroll up?
second, can you scroll up?
Yeah. So when light fall on the leaf, oxygen produced. Other than oxygen, water break, electron transport. Because of electron transport, we get all our product. If somebody asks, summarize the light reaction summary will be when you consume light, you take photosystem. PS means photosystem. Okay. and you take water, correct? In case of chlorophyll, because we need chlorophyll in case of light reaction, what will happen? There will be production of ATP, NADPH, oxygen, and hydrogen. These hydrogen are really crucial for the ATP formation, correct? But hydrogen will not be there in the final reaction. It will be intermediate. We'll study in the chemiosmotic hypothesis. Any doubt? This is the summary of the light reaction. In this light reaction, we have last topic, chemiosmotic hypothesis. As the name suggests, osmosis. Osmosis means movement of something from high concentration to the lower concentration. And chemi means chemical. Normally, chemiosmotic hypothesis. Normally, osmosis occur for water. But here, we are not using water. Osmosis definition is movement of water from high water to less water.
Sir, I'll join from my phone. My mom needs the laptop.
Sure. She joined.
She'll take like 2 minutes to join, sir.
I'll come in 2 minutes. So, chemiosmotic hypothesis. लास्ट ईयर मैंने बहुत बार समझाया था ऋषिकेश एंड रेयान. डू यू रिमेम्बर? So many time I taught this topic with the same story, and
Yes.
people
So
were
that's
like, कि सर
absent.
स्टोरी तो समझ में आ गई, टॉपिक समझ में नहीं आ रहा है. एनसीईआरटी से रीड करने के बाद.
उसी दिन मैं एब्सेंट थी सर, मुझे अभी तक याद है.
अच्छा, बहुत बढ़िया. So, chemiosmotic hypothesis. Osmosis means movement of water from high water concentration to low water concentration with the help of the semi-permeable membrane. This is the definition of the osmosis. Chemiosmosis means movement of hydrogen, chemical, and here you have chemical hydrogen. So movement of hydrogen from the high H positive concentration to low H positive concentration with the help of the semi-permeable membrane. With the help of the semi-permeable membrane. Okay. And which semi-permeable membrane are we using? We are using thylakoid membrane. Through the thylakoid membrane, hydrogen will move from the high H positive to low H positive. What is the benefit? Whenever, example, you have this thylakoid membrane and this is semi-permeable membrane. So when hydrogen pass from the high H positive inside, low H positive outside. So when this thing happen, what will happen? ATP will produce. Okay, ATP will be produced. ATP will be there. Any doubt? We want to understand how we have to increase more number of hydrogen in thylakoid lumen. I will write lumen only. Thylakoid lumen, and this is stroma. So ultimate purpose is we have to increase more H positive inside the lumen, thylakoid lumen, and we have to decrease stroma. What are the different methods for this? First method is water splitting. If H2O break, you know this H2O will break down into the H positive ion plus oxygen plus electron, correct? So it means this water splitting process should happen on the inner side of the membrane. Why? Because H positive should produce inner side. Any doubt, guys? First step in chemiosmosis. Second, this water splitting is associated with PS2 only. On the inner side, what will happen? H2O will be broken and H positive released. So there is more H positive concentration. Correct? Second, you know H positive should reduce from the stroma. How to reduce from the stroma? Example, when electron move from PS2 to PS1. Remember, in chemiosmotic hypothesis, we are revising the same PS2, same, what you said, non-cyclic photophosphorylation again. Here you have PS1. So in this PS1, you know NADP will convert into NADPH. So what happened actually in this case? Hydrogen are added in NADP. What is the source of this hydrogen? Stroma. It means this NADP should be located on the stromal side. So what is the benefit of this? Hydrogen from stroma will reduce, and because of the H2O, hydrogen inside the lumen will increase. So there will be gradient known as proton gradient. Here you will have proton gradient. High H positive inside, low H positive outside. So this is proton gradient. If this proton gradient break, ATP will produce. Okay. Here to transport the electron from one system to another system, you have cytochrome B6F. You have cytochrome B6F. Okay. So there are another process. There is a special structure present on the outer side, plastoquinone. Plastoquinone. This plastoquinone will accept hydrogen from the outside stroma. Again, we are reducing hydrogen from the stroma because you want to transport electron. There is a story I told last year. You have watched this movie Three Idiot, right? All of you.
Yes.
Yes, sir.
Ravi. Huh? बात कर लो फिर एक बार. Hello. So guys, look at this. In ultimate purpose in this topic is to increase H positive ion inside the lumen and decrease H positive ion from the stroma. Correct? Water help to increase the H positive ion and NADP help to reduce the H positive ion from stroma. Correct? Even when you want to transport electron, when you want to transport electron, there are two very popular structures. From PS2 to cytochrome B61F. This is neat PYQ, by the way. You have a carrier that transport electron, and that carrier name is plastoquinone. This plastoquinone also accept as positive from the outside, and when this PS2 transport electron, what will happen? It gain of electron is what? It will gain electron. So after gaining hydrogen and electron, what will be its nature? It's plastoquinone. Tanvi, tell me in chemistry, alcohol, phenol, ether, if you take ketone and it undergo reduction, what should form?
Sir, secondary alcohol.
Secondary alcohol, correct? So alcohol should form. Now tell me in plastoquinone. So plastoquinone means you have ultimately ketone. You
It's a
accept
secondary ketone. I
as
mean,
positive
secondary
from
alcohol.
stoma, and so it will be plastoquinol, secondary alcohol, from ketone
Yes, sir.
to alcohol,
Yes,
correct?
sir.
So it will convert into plastoquinol when it again convert into plastoquinone, hydrogen will be produced inside, correct? Here actually same thing happening. It is plastoquinol. When again convert into this, hydrogen will be produced inside and electron is passes further. Any doubt? I told you story of this 3 idiot. There is a guy Chatur, right? And this Chatur have one philosophy, though it is bad, but this is how he works.
Distract other
Either,
people instead of studying yourself.
yeah, either you study well and get the first rank, or you distract other people so they don't study, automatically you are the topper. Same thing here. Either you increase as positive concentration in lumen, or you decrease as positive concentration of stroma, so automatically lumen will be high, correct? Now this is the process. When you transport electron from PS2 to PS1, plastoquinone is saying ki, no, I don't transport electron, I transport only hydrogen, I need hydrogen. So it's basically bribe. Plastoquinone ki, I need money, only then I will transport your material. So when we want to transport electron, plastoquinone alone cannot transport only electron, so they need hydrogen. So hydrogen will be transported inside. So imagine ऋषिकेश, अगर यहाँ पे प्लास्टोक्विनोन हाइड्रोजन को एक्सेप्ट कर रहा है और अंदर ट्रांसपोर्ट कर रहा है, तो प्लास्टोक्विनोन इनर साइड होना चाहिए या आउटर साइड?
सर आउटर साइड में होना चाहिए।
वेरी गुड, आउटर साइड। तो क्योंकि वाटर की जब स्प्लिटिंग हो रही थी, सो वाटर स्प्लिटिंग वाज रिस्पांसिबल फॉर मोर एच पॉजिटिव कंसंट्रेशन, सो इनर साइड. बट प्लास्टोक्विनोन, because it accept H positive from outside, so it should be outer side logically, correct? Next, from cytochrome B6, now we have to transport only electron, no hydrogen transportation. For this we have plastocyanin. Okay, from cytochrome B6F to PS1, you have electron carrier plastocyanin. So Anvita, tell me, plastocyanin have to transport only electron, no hydrogen. Should it be inner side or outer side? Anvita,
Sir, can you repeat plastocyanin?
should be inner side or outer side?
Outside.
इनर साइड, क्योंकि आउटसाइड के लिए कुछ है ही नहीं ना वर्क करने के लिए,
अच्छा.
है ना? ओनली इंटरनली इलेक्ट्रॉन हैव टू ट्रांसपोर्ट, सो इट कैन. एंड नाउ प्रतिज्ञा, Tell me, in PS1, we have to accept hydrogen from the stroma. So should it be outer side or inner side? NADP reductase. Pratigya, is she there?
Yes.
Pratigya. Barinder, it should be outer side, stromal side. Why? Because from stroma it have to accept as positive ion. Outer side, stromal side. Correct. So these wording are extremely important. Inner side or outer side. So logically, if you are correlated with the external work like plastoquinone and this NADP reductase, both have to accept as positive from outside. That's why outer side remaining are inner side. Done. Understood? But as of now, you have whatever. Your ATP is not produced. And ultimate goal to understand this process is ATP formation. Remember, when I explain the significance of this topic, is that when we understand how photosynthesis occur in plant, we can manipulate the photosynthesis. When nowadays we are growing crop, any crop, we want to increase production. Production means increase photosynthesis. How we can increase photosynthesis? By adding some fertilizer, correct? We are adding some mineral. What is the role of all these artificial things? To increase the production, correct? Because you can ask, sir, even if we don't read about the photosynthesis, plant will naturally do photosynthesis. Yes, this is true. But when we learn about this, we can increase the production. So the kind of population we have in the world, we need more food. Correct? We need more food. That's why. So last part. In this chemiosmotic hypothesis, yes,
Can you scroll up, sir?
sure. So let me tell you what could be the question asked in this. Um, Water splitting associated with which PS? PS2. Is water splitting inner side or outer side? Inner side. Where plastoquinone located between PS2 and cytochrome B6F? What is the role of plastoquinone? It convert into plastoquinol, transport hydrogen along with the electron. Where it is located outer side. Plastocyanin between B6F
Yes.
and PS1. Correct? So what are the things required in this chemiosmotic hypothesis? Definitely we need membrane, semi-permeable membrane, correct? We need pump to transport the hydrogen. We need the proton gradient. So if you want to produce ATP, you need proton gradient high as positive to low as positive. You need ATP synthase enzyme for the synthesis of ATP. Correct? So these are the important things what you need for the chemiosmotic hypothesis. Correct? So now look at the dam example. This is your thylakoid membrane. Okay, thylakoid membrane is like your dam. Dam have important channel, okay, gate channel, and this channel have a turbine also. Outer part is this is turbine. This is channel. Channel means gate opening, and this is turbine. And when turbine rotate, electricity is produced. Same here. When this is known as CF1, CF naught. Okay. So you already have high as positive concentration inside. and low H positive concentration outside. So when hydrogen pass from inside to outside, it should pass through the channel, correct? And this channel is integrated inside the membrane. But when same water hit on the turbine CF1, what will happen? Conformation change will happen, means turbine will rotate. In biology, we write conformation change, but in reality, it is basically change in the shape of the CF1. When it change, what will happen?
So can you say what
ATP
is
formation.
CF1 and CF naught?
CF0 and CF1 are basically the system of passage of water. Example, you have a dam. Dam have lot of water, right? A lot of water compare with the H positive ion. You have lot of H positive ion. When you open the gate of the dam, what will happen? Water will come out of this same way as positive will come out of this. Then this water from the dam will hit the turbine, correct? Turbine will
हाँ
rotate
सर
and it produce electricity. Yes, Pratigya?
सर मैं थोड़ी देर में आती हूँ
ठीक है. So as positive when hit the turbine, it will rotate and it will produce electricity. It will produce electricity. Same way, when as positive pass through this, first it will rotate the turbine CF1 and it will be responsible for ATP formation. What is the purpose of this chemiosmotic hypothesis? How ATP will produce. How ATP will produce. In the presence of this whole thing together is known as ATP synthase enzyme. ATP synthase enzyme. Don't write ATP age. ATP age is to break down ATP. ATP synthase to produce ATP. Any doubt, guys?
No, sir.
And
Yes,
if you
sir.
see question from this topic are very easy and very straightforward. You will see this on 3rd May 2 p.m. Correct, Jordan?
Yes, sir.
Rubisco enzyme. First question.
So I'll be back in 5 minutes.
Okay, Jordan. So, Rishikesh, which of the following is required for the light reaction of the photosynthesis? Do we need CO2 for light reaction?
Yes, sir.
No. Like we
Uh,
use as of now anywhere CO2 in light
yes,
reaction?
sir, yes, sir. For light reaction, we don't need.
We don't use. So A cannot be there. Oxygen is required for the light reaction?
No, sir.
No. Oxygen is produced, not
Yes,
required. H2O is required for light reaction?
yes, sir.
Yes. H2O is required because it is associated with PS2. Water splitting happen, oxygen produced. Chlorophyll,
Yes, sir.
light.
Yes, sir.
Of course, light reaction, light.
Yes, sir.
So, what is
O2
the
is the
answer?
byproduct, we can say, right?
Yes, Ryan?
So O2 is the byproduct, we
Yes,
can say.
O2 is product of light reaction, not required for the light reaction. So, can you say
Yes.
answer will be B?
Yes, sir.
The
Answer will be sir A and B only, sir.
no. A and B are not there. C, D, E will be the
Ah,
answer.
yes, sir. Correct answer. So CD,
Uh,
sir.
uh, see, Ryan, you asked the same question. Product that come out of the chloroplast diffuse out.
O2, sir.
Done.
So what is the diagram?
Diagram will discuss. Three cycle.
Yes, sir.
Tanvi, this question. I will check your answer. Write your answer in the chat box. I will read everybody's answer. Reaction
So,
center in PS2 have absorption maxima at
so PS2 will be 680.
and PS1.
It's 700.
Yes. So 680. How many wrote answer? 5 people. Rayan D. Barinder D. Only 2 people wrote answer. Next time, everybody write. Jordan, are you there? Sarah, are you there? Sarah, not there. Then Nirjala,
Yes, sir.
answer for this question.
D option.
Perfect. Done. So for chemiosmosis, we need membrane, thylakoid membrane, proton pump, proton gradient, and ATP synthesis. How many of you answered this question? Ryan, Vrinder, Nikki, Nirjala, D. Okay.
सर वो मैंने तन्वी को डायरेक्ट मैसेज किया था तो वो हटाना भूल गई तो वो आंसर उधर ही रह गया तो वो मेन चैट में गया नहीं
कोई बात नहीं. अभी लिखना. But let me check how many of you are there. I want to see everybody to answer. Total 11 people. 10. Aditya, Anvita, Pratigya, Varinder, Manan, Jordan, Nikki,
Yes,
Nirjala, Rayan, Tanvi, and Rishikesh. Chandana came.
क्या कर रही हो?
Manan, 14th question. Incorrect. Grena lamla means grena.
Green.
Your answer.
D.
Answer will be 2. Cyclic
Yes, sir.
phosphorylation have only PS1, not both.
साइक्लिक वाली अच्छा वो वाली कह रहे हो आप जो इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम जिसमें आएगा बीच में मतलब घूम जाएगा जो क्लोरोफिल पे ही वापस.
Exactly. Second thing, guys, grena lamella means non-cyclic. Stroma lamella means cyclic. In grena lamella non-cyclic, both PS1 PS2 present. In stroma lamella cyclic process, PS2 is absent, NADP reductase is absent, only PS1 will be present. Let me see how many of you wrote the answer. 5 people. Nikki B, Varinder B, Dayan DB, Varinder D. Only 3 लोगों ने.
Stage in the chat box. I don't know why.
Okay, so you can answer directly.
Okay.
Right. So remember, grena lamella means non-cyclic process. Stroma lamella means cyclic process. Tanvi.
So first one is correct.
Correct.
Second one is also correct.
Correct.
Third one.
Let me tell you third one. It is talking about the primary acceptor of electron. Primary acceptor of electron from PS2 is plastoquinone. And plastoquinone is a hydrogen carrier, not electron carrier.
Yes, sir.
Plastoquinone is a hydrogen carrier, not electron carrier.
Plasmaphenon is you said that hydrogen carrier. It carries hydrogen, but it also carries the electron, right? It transfers it to the
Yes, but
cytoplasm.
it will transfer electron only in the presence of hydrogen.
Okay. Means if hydrogen is there, then only it will transfer.
Correct. So it is main thing is the hydrogen carrier. Next, Tanvi. Ryan is also not answering in the group, so I will ask her directly. Next question, Ryan.
Um, so C is wrong. So the answer will be like NADPH reductase enzyme is located on the stroma side of the membrane. Correct. So the answer will be C, sir.
Let me see what other people. C is the right answer. Let me see how many people wrote 4. Ryan C, Chandana C, Brinder C, Anita C. Answer will be C only because proton increase in thalacod lumen, not in stroma. NADP reductase outer side because it have to accept hydrogen from stroma. Primary acceptor of electron is plastoquinone that transfer electron to the hydrogen carrier. It is hydrogen carrier plastoquinone.
So then the proton increase in the like the inner side, right? Not
Yes,
stroma.
in the
The
lumen.
grana.
Yes,
Lumen, sorry.
lumen of thalacod. Like B
Lumen.
statement is correct. So definitely E is wrong if B is correct. This Ryan. Not true regarding the release of ATP during chemiosmosis. What about first statement?
So like it's not true.
If it is proton gradient, it cannot be electron gradient. So answer will be
Yes,
B.
yes, sir.
Correct. NADPH stromal side correct. Proton movement across the membrane to stroma correct. Proton gradient happen correct.
So A is correct. C is also correct. So D1 also correct. So maybe option B.
Yes, B is wrong because if it is a breakdown of proton gradient, how it can be electron gradient? Answer will be B. Let's see how many people wrote. Nirjala. Only Nirjala.
Sir,
Why
I'm
other
not
people?
sure of the answer.
So I
Yes,
said you.
Anita. So I hope all of you understood this concept properly. Chemiosmotic, non-cyclic, and cyclic. Raise your hand if you feel yes, sir. Everything perfect, no issue. Nirjala, Aditya, Nikki, perfect. Brinder, Ryan, Chandana, Anvita.
Yes, just more questions, sir.
Yeah, we need to do more question
Because
for sure.
the questions, if the question is tricky, then it would be a little difficult, I guess.
Hmm. Hmm. Correct. Then what about Jordan? You are there? Jordan, Manan, ऋषिकेश समझ में नहीं आया ऋषिकेश
आ गया
I just came back, sir.
ओके मनन ओके जॉर्डन ऋषिकेश
सर मैं पानी पी रहा था एक्चुअली
ठीक है कोई बात नहीं ऋषिकेश यू आर देयर
शायद वो गंगा नहाने गया है ऋषिकेश
हाँ so this topic is done and if this topic is done it means light reaction is done. As I mentioned, light reaction means in this process you consume. Oh, we did this question also, and very first question was this only. What is the conclusion? What is required in the light reaction? That was the question. Light, H2O, and chlorophyll. This was the question, right? If next time ask what are the product of the light reaction, these three are the product of light reaction. Correct. During this process, because oxygen is produced, oxygen will come out. Oxygen come out. Um, there will be utilization of the remaining ATP and NADPH. These two are utilized for food formation, food synthesis. Okay, that's why it is also known as carbon fixation. Why? Because carbon is utilized, CO2 is utilized. CO2 is utilized. That's why this biosynthetic phase is also known as carbon fixation. Carbon fixation. Carbon fixation. Okay. If carbon fixation happen, what will happen? Yes, Ryan.
So what is this you have written here?
मुझे पता
The
था. Carbon fixation.
okay.
Carbon fixation or biosynthetic phase because food is produced. How food is produced is explained by the Kelvin. Kelvin gave the complete process how food is produced by using algae plant using the radioactive carbon-14. By using this radioactive, he explained the full mechanism of food formation. Full mechanism of food formation, that is Kelvin cycle. How glucose is produced is the process. So what happened? I will give the simplest cycle. When CO2 is utilized, then it will react with 5 carbon molecule known as RUBP. So 5 carbon RUBP, 1 carbon CO2, total 6. Total 6. So 2 molecule of 3 carbon will produce PGA, phosphoglycerate. 2 molecule of 3 carbon PGA is produced. Correct? You will be surprised, ki, sir, if 3 molecule product is produced, then reactant should contain 2 carbon. Why? Because 1 CO2 will be added. But don't worry, we have studied a lot and this is correct. And RUBP contain ketone in this. Okay, it contains ketone in this. And you need enzyme for every process. Enzyme in this case is Rubisco. Why? Because here you can add CO2, that's why carboxylase. You can add oxygen, so oxygenase. That's why Rubisco, one of the most abundant enzyme in the world. Or most abundant protein in the world, right? This step is known as carboxylation. Why? Because addition of CO2. Carboxylation. Okay. Addition of CO2. Then this PGA will undergo change into 3 carbon only, and this process is known as reduction. Reduction of what? Reduction of PGA. Kaise? Because in this case your NADPH will be used and convert into NADH. Sorry, NADP. Added DPH from into NADP. So what is happening? This NADPH is producing hydrogen. This hydrogen is utilized. This hydrogen is responsible for reduction of PGA. Remember, reduction of PGA, not reduction of NADPH. Reduction of PGA. Reduction of PGA. Reduction of PG. Also, ATP is utilized here. ATP and NADPH both are utilized. Why? For food formation, you need energy. Correct? ADP is produced. From this, you remove one carbon, two molecule. If you remove one carbon, you will again form RUBP, 5 carbon. Correct? You regenerate same thing. That's why this step is regeneration. This step is regeneration. So, if you see, this is the first step, carboxylation. This is second step, reduction. This is third step, regeneration. Correct? And then RUBP again utilized. Right? Even in regeneration, you know one ATP will be consumed. Jordan, do you remember I told you this story? If you want to get money back, regeneration of your own money, do you think you will easily get? Example, you have to take money from the Tanvi. Do you think you will easily get?
No, not at all. Very tough
So
job.
you have to invest energy to get money.
Yes, sir.
That's why regeneration process require ATP. It
So he's
require
telling that I give him money.
you give him money. So Tanvi, tell me, if you give him
Sir,
money,
she's broke. She always asks me for money.
Shut up, Bob. Replay it every single time.
Oh, please.
okay,
She keeps
okay,
running to me for sandwiches,
wait,
sir.
wait, wait, wait. So Tanvi, just imagine, if he's not giving you money back, what will you do? What you will use your hand and leg?
Sir, I
ATP.
just see the thing is we've been like I don't know who has to pay who back right now.
Okay. No, but in case you need and he's not giving,
Sir, I'll kick him, sir.
exactly. Kicking require energy,
Yes,
ATP,
sir.
muscle contraction,
Yes, sir.
right? So
Yes,
same
sir.
ATP is required here. Understood, everybody? So that's the thing. If you see in one cycle, so if you see in case of one Kelvin cycle, in one cycle, what happened? You used one CO2, correct? RUBP is regenerated. So this CO2 will be used to produce glucose, correct? But one is not sufficient. You need six CO2, correct? In one cycle, one CO2 used. How many NADPH? Two NADPH is used. How many ATP? Three ATP is used. Done. So one CO2, two NADPH, three ATP in one cycle. Example, if you want to produce glucose, you know you need six cycle for this. Here you need six cycle. Correct? If you need six cycle, what will happen? In that case, we need six CO2. Double of 2 is 12 NADPH and 18 ATP. Tell me, any doubt? Basically, if you want to produce food, glucose, you need 6 Kelvin cycle. In 6 Kelvin cycle, this number of NADPH 12, 18 ATP and 6 CO2 is required. Required, need. Correct? Done. This topic is done. What happened in the Kelvin cycle? 3 step. Carboxylation where CO2 is added. Reduction where PGA will undergo reduction by addition of NADPH and ATP. And last again RUBP regenerated by the investment of ATP. If you see what is the CO2 acceptor, CO2 acceptor is this. What is the first product? First product of the Kelvin cycle, PGA. Correct. First product is First product will be PGA. If you understood this well, we saw some question of 1880p and all this stuff. Barinder, answer of this question. When one CO2 is fixed as one molecule of triose phosphate, which of the following made high energetically intermediate use in the reduction phase? In the reduction phase. Reduction phase.
A.
A. Very good. A is the answer. कई बार हमें लगता है कि अच्छा in 1 Kelvin cycle there are 3 ATP and 2 NADPH. Options are not good in this question. Options are not good. They should have given 3 ATP and 2 NADPH. A is correct. Why? In reduction step, you can see one PGA require one NADPH, so two PGA require two NADPH. One PGA require one ATP, two PGA require two ATP. So in this reduction phase, you need two NADPH and two ATP. This is the answer, but options are not good.
So why you said options are not good?
Because if they give 3 ATP and 2 NADPH, then you will be confused. Here it was straightforward.
Then so if it is given 3 ATP and 2 NADPH, then also the answer would be 2 ATP and 2 NADPH, right?
Yes, but people will think because they read so much ki in 1 Kelvin cycle there are 3 ATP and 2 NADPH, so they will try to mark that also.
Yes, sir. Yes, sir.
So Jordan, 9th question. Everybody, I'll check answer of this question in your group, like chat box. ठीक है. Okay. ठीक. Next class study session 12:30. क्लास हम रखेंगे 4.
Like the study
See,
session is from 12:30 to 2:30, right?
yeah.
And so 4 by you say.
Huh?
So I may have to
12:30
go
to
outside,
2:30.
sir.
What, Jordan?
Sir, I may have to go outside, sir.
Omni has to go outside. Anvita
Like our
A,
exam
Ryan
schedule
A,
is the same
Tanvi
till
A,
now.
Nirjala A, Nikki A, Chandana A. So 1880p NADPH. Now, guys, are you getting the point? Keep chapter look difficult, but questions are easy.
So the problem was we didn't understand the topics, I guess.
Okay. Now you understood everything?
Yeah.
Okay. So, next, last part and the most important popular topic is C4 cycle. In C4 cycle, you have to compare with what happened in the C3 cycle. Remember, in all plant, C3 is must. C3 always present. Okay, C3 always present. Whenever C4 cycle happen, then C3 is definitely part of this process. Okay. In C4 plant, only difference is in case of C3, CO2 was reacting with RUBP. In C4 plant, CO2 will react with the phosphoenol pyruvate, three carbon molecule. Phosphoenol pyruvate. Phosphoenol pyruvate. When both react, then it produces the first product, oxaloacetic acid, four carbon product. Correct? In C3, three carbon product. C4, four carbon product. In C3, acceptor is 5 carbon. Here, acceptor is 3 carbon. Enzyme in C3, Rubisco enzyme in this case is peptidase. Phosphoenol pyruvate carboxylase, peptidase. Correct? All these names are important. Comparison is extremely important. Then this oxaloacetic acid convert into malic acid and then aspartic acid. Okay. All these things what we have studied happen in the mesophyll cell. Mesophyll cell. Even what we studied here in C3 cycle happen in mesophyll cell. Mesophyll cell. Correct. But there are special cell in case of C4 plant. You will have bundle seed cell. You will have bundle seed cell. Okay. In bundle seed cell, there is a connection between mesophyll and bundle seed cell. Like this. Okay. So this is the connected. So aspartic acid will come here. Decarboxylation happen because you have four carbon product. So CO2 will be removed. This CO2 undergoes C3 cycle again, and three carbon molecule is also produced. This three carbon molecule will go back because regeneration happen. Subchangasi again. And regeneration of phosphoenol pyruvate. Done. This is bundle sheath happen in bundle sheath cell. This happen in bundle sheath cell. So what is difference between bundle C3 and C4 plant? C4 plant can tolerate more temperature, can tolerate the extreme condition. Productivity is high. Number of chloroplasts in each cell is high. Other difference are Rishikesh. Okay. Then you have question?
Yes, sir. So can you just say again that the bundle sheet cell one?
Bundle sheath cell are the special cell present in the C4 plant. In C3 plant, everything happen in the mesophyll cell. Example, in C3 plant, Calvin cycle occur in mesophyll cell. But in C4 plant, this same Calvin cycle occur in bundle sheath cell. Bundle sheath cell, correct? Example, bundle sheath cell have Rubisco because Calvin cycle occur in case of bundle sheath cell, correct? But in case of bundle sheath cell, pap cage is absent. Pap cage absent. In mesophyll cell in C4 plant, mesophyll cell, pap cage is present. Pap cage present, but Rubisco absent. Any doubt? Very, very, very popular. So you should know this pathway is known as Hatch and Slack pathway. Scientist name Hatch and Slack pathway. And C4 plant are like maize, sugarcane, sorghum, bajra, correct? So all these are the simplest C4 plant that can tolerate high temperature. You can see ki all these plants mostly occur in the area where you have water shortage. Any doubt? And difference is really, really, really important. In C3 plant, in C4 plant, what are the difference? Difference is extremely important. If you don't understand difference, trust me, you don't know this topic. Example, in case of C3 plant, if you talk about cell, so here you have mesophyll cell. Here you have mesophyll cell plus bundle sheath cell. Correct? Then in C3 plant, primary CO2 acceptor, who will accept this CO2? Here C3 plant RUBP. Here phosphoenol pyruvate. Enzyme in this process, in acceptor process. Here enzyme is Rubisco. Here enzyme is peptidase. Correct? Then you know, in case of first product formed in the process. In C3 plant, PGA phosphoglycerate is formed. Here oxaloacetic acid is formed. This is 3 carbon, this is 4 carbon. Difference is important. 3 carbon, 4 carbon. Then what are the other difference? Temperature tolerance. Temperature tolerance is more in C4 plant and less in C3 plant. Mostly these act in 20-25 degrees Celsius. This can tolerate up to 40 degrees Celsius temperature easily, 40-45. No. No problem. So, so what else? If you take, Rubisco. Rubisco enzyme are present in the mesophyll cell in C3 plant and bundle sheath cell in C4 plant. Correct? There is one very popular term, photorespiration, kind of waste process, photorespiration. It is present in C3 and absent in C4. Tell me up to here any doubt? Up to here any doubt, guys? How many of you understood C3, C4 cycle thoroughly?
Me,
Raise
sir.
your hand.
So can you show the screen once?
Yes. Aditya, Rishikesh, Anvita, Jordan, Barinder. Chandana, Ryan. What about Manan, Tanvi? Manan and Rishik is already
सर
there.
आ
Manan
रहा है.
आ रहा है, धीरे धीरे आ रहा है क्या?
हाँ मतलब आ तो रहा है, बस ये कि थोड़ा सा खुद भी पढ़ना पड़ेगा, रिवाइज करना पड़ेगा, तब
हाँ
होगा पूरा क्लियर.
हाँ, रिवाइज करना पड़ेगा, लेकिन क्वेश्चन के थ्रू पता चल जाएगा कि मोर और लेस एक ही टाइप के क्वेश्चन आते हैं.
हाँ मोर और लेस सही हो रहे हैं, मतलब कोई एक आधी गलत जा रहा है.
हाँ. So if you are done with this, let's do 3 4 questions, and you will feel कि yes, we know the topic. Majority part of this chapter done. Like 95% of question asked what I taught. 95% question of this chapter. Hardly 1 2 from the factor affecting the photosynthesis or scientist name. Both are very easy and straightforward. No understanding required in factor affecting the black man is important. Black man in 1905. If you are done with this, see question. I will show you from the beginning. Very first. On this part, we'll do later. Mm. Rishikesh, which of the following is true about Rubisco?
Yes, sir. It is the catalysis of water. It catalyzes the oxidation of to
Everybody write answer in chat box. Rubisco is the most popular enzyme. Rubisco can bind with CO2 and oxygen both. But if you ask, sir, which one? Which one have more affinity with Rubisco, CO2 or O2? Answer is CO2. Very high.
CO2.
If you have more CO2 and less oxygen, Rubisco will bind with
Yeah, I think the option B is correct, sir.
option B is correct. Yes. So let me write this. So if you talk about Rubisco, then Rubisco can bind with both CO2 and oxygen, but more affinity with CO2. More affinity for CO2. It means if you have more amount of CO2 compared to oxygen, definitely it will go with CO2. If you have equal amount of oxygen and CO2, it will bind with CO2. But if you have very high pressure of oxygen, like more availability of oxygen, then no choice. Some, not fully, some bind with the oxygen. Some amount of Rubisco will react with the oxygen as well. And reaction of Rubisco with oxygen is photorespiration. Why? In this case, if you see Rubisco means basically RUBP, 5 carbon molecule combine with oxygen. So PGA will form as usual, but 2-phosphoglycolate will also form, very popular. 2-phosphoglycolate is also formed. This is 2 carbon. This is special. Why? Because in normal C3 cycle, Calvin cycle, 2 PGA molecule were formed. Here only 1 PGA molecule. Why? Because total carbon is 5. 3 plus 2, 5. Correct? Total carbon is 5. 3 plus 2, 5. But if you see, last time there was CO2 addition, 1 extra carbon. That's why it was easy. Second, this is a waste process. Why? You know, in case of this photorespiration, why it is waste process? Because no ATP produce, no NADPH produce, no food produce. Instead, there is a wastage of ATP. Wastage of ATP. So wherever photorespiration process present, productivity will be. Photorespiration means it will reduce productivity. That's why it is present in C3 plant, absent in C4 plant. Anybody have doubt?
No, sir.
Hmm. Higher affinity with for oxygen is wrong.
So can we scroll up?
Scroll up.
Yes.
Where?
So not the PDF.
Yes. So with this majority part is done. This color is pending. Color of chlorophyll A is blue green. Color of chlorophyll B is yellow green. Color of carotenoid is carrot. Carrot means orange. So orange color. And color of xanthophyll is yellow. Color of xanthophyll is yellow. So remember in this case, A main color is blue because in blue region and red region maximum photosynthesis. Out of these two, blue is more. So maximum photosynthesis in the blue region, correct? So blue region means chlorophyll A is bluish color, correct? Carrot, orange color. Carrot is orange color, correct? Chlorophyll B is still green and xanthophyll yellow. Done. You can solve this question, correct? Carrot orange, A bluish color, correct? Chlorophyll B is still chlorophyll. Chloro means green, so have greenish color. And last yellow left, xanthophyll. Any doubt? Any doubt? Color of these four. Two times same question asked. So that's why it is important. And we'll do this if you don't understand. Okay, guys. So have a break for half an hour, 12 to 12:30. From 12:30 to 2:30, you have a break. Sorry, study session. In study session, for everyone, I will give different tasks. The moment you enter into class, turn on your video and ask, sir, what I have to do. Or if you have your plan, you can say, "Ki, sir, this is what I am doing. I will ask you question at the end of the session. Every single person. Whatever you are doing, you can ask me or you can tell me, ki, sir, this is what I am doing. At the end of the session, I will ask question. Is it okay for everyone?
Yes.
Yes, sir.
Yes, sir.
And I hope now you don't have any issue in the photosynthesis
Yes.
chapter anymore.
Yes,
But
sir. No issue. Just have to.
but make
So can you
make
send the
sure,
PDF in the group?
yes, I will send PDF in the group and make sure in study session at least half an hour solve at least 50 question to have clarity. Then you can go through your test or full syllabus so you will get idea. But read solve 50 question to have better clarity and ask question during the study session.
Yes.
Yes,
I
sir.
will send I will send this PDF in the group.